Hello and welcome to today's lecture, which will serve as an introduction to adaptive immunity. Now, previously you've heard about innate immunity. So, upon exposure to a pathogen or any sort of antigen, at first your innate immune response is launched that is very not pathogen specific. So, these are responses that would be triggered upon any sort of infection. After a few days, however, if the innate immune system is unable to clear the pathogen or the antigen that is infecting the host, then the adaptive immune response is launched. The adaptive immune response is highly pathogen specific, which we'll go into the details of throughout this lecture. Now, if you look at the course of the infection from the point of view of a pathogen that you've seen before, which is also, you can look in your book at figure 2.1, you can see that there's a range of different immune responses against pathogens that'll range from being able to target lots of different types of antigens to only one specific pathogen, and so the pathogen specificity goes from very low to very high. Now, previously, you've learned about innate immunity, which is this very broad response to a variety of different pathogens, and today we'll be focusing on adaptive immunity. Now, adaptive immunity, as I just mentioned, occurs when the innate immune system is unable to clear the pathogen that's infecting the host, and thus triggers B and T cells to recognize the pathogen for which they're specific, hopefully ending up in the removal of the infectious agent. Now, in today's lecture, we'll be going through a very brief introduction to lymphocytes, which I think you are already a little bit familiar with, but it'll serve as a reminder. Then we'll go through what's called the clonal selection hypothesis, followed by a discussion of antibody and T cell receptor structures and how they differentially recognize antigens. We'll then talk briefly about MHC molecules and how MHC molecules and T cell receptors interact. And we'll end by a very brief discussion on how the adaptive immune system is actually activated by the innate immune system. Now let's start with an introduction to the key players in adaptive immunity. So this is a figure that you guys are all very familiar with, which shows how the development of the various immune cells occurs from the pluripotent hematopoietic stem cell in the bone marrow, which allows for the generation of both innate immune cells, which we've talked about before, as well as adaptive immune cells, which we'll discuss today. Now, the lymphocytes we'll be discussing today are T cells and B cells, and these serve as the most active players in adaptive immunity. Very general properties of lymphocytes include that they're highly specific for a unique epitope, and we'll go into what that means in a little bit. They manifest very little functional activity in the immune response until they encounter the antigen for which they are specific. And after activation, lymphocytes form memory cells, which are very critical in enabling a host to have a very long-aged memory response to specific antigens. Now, diving a little bit deeper into what our two players are of lymphocytes, we have our T cells, which look like this, where they have their T cell receptors on their surface of their cells. And we also have B cells, which have their receptors also on the surface of their cells, but they also secrete their receptors in a secreted form called antibodies. Now, T cells are derived from the thymus, which if you remember your development lecture, we went through that, but if you would like to review, going back to the lecture entitled Cells and Organs of the Immune System would be a great place to go for reference. And we also have our B cells, which were originally named for being bursa-derived cells, so the bursa is an organ in birds, but by happenstance, a bone marrow where B cells differentiate also starts with B. So bone marrow or bursa is the way to remember B cells. Now, T cells mediate cell-mediated immunity, and B cells mediate humoral immunity or antibody-mediated immunity. And T cells come in two very broad flavors. First are CD4 positive T cells, or T helper cells, which help in the stimulation and differentiation of the immune response, and also CD8 positive T cells, which are cytotoxic T cells, which will directly kill infected cells. Now, I realize I just gave you a lot of information, but this information will review throughout the remainder of the lecture, and it will also be touched on in future lectures and future concepts. Now, the clonal selection hypothesis, which we'll be going through now, is really important because it really outlines why the adaptive immune response is so critical in being able to respond to antigens. The clonal selection hypothesis was originally proposed by Macfarlane Burnett in the 1950s, and it's really the central paradigm of adaptive immunity. And it states that specific lymphocytes proliferate in response to the antigen for which they are specific, and subsequently they differentiate into antigen-specific effector cells to eliminate the eliciting pathogen. That is a lot of words, as most hypotheses are, so let's dive into this step by step of what the clonal selection hypothesis is. First, we know from the development of the immune system that we have a single progenitor cell, a hematopoietic stem cell, that gives rise to a large number of lymphocytes. And each of these individual lymphocytes have a single type of receptor for which they have a unique specificity. So for example, here, this progenitor in yellow without a receptor leads to the 7 different colored lymphocytes that have a unique specificity for a different antigen. Next, the clonal selection hypothesis states that self-reactive immature lymphocytes, so these are lymphocytes within the bone marrow or the thymus, respectively, of what cells they become, these will be deleted at very early stages of their cell development, such that mature lymphocyte receptor repertoires lack the receptors specific for ubiquitous self molecules. So if we go back to our example of the 7 lymphocytes that derived from that original stem cell, we can see that 4 of these 3 are reactive for self-antigens. This means during development, these cells have receptors that would react against proteins that are found within the host. To prevent autoimmunity, these cells are eliminated during development, such that only the cells that are not reactive against self-proteins can leave the bone marrow or the thymus. So in this example, just these 3 central lymphocytes would be able to leave the organ of their development to go into the rest of the body. Next, we know that mature naive lymphocytes can interact with a unique antigen. Now, I want to remind you that mature naive lymphocytes are those cells that have left the organ of their development, so the bone marrow or the thymus, that have not yet interacted with their antigen, which means they're naive. So these cells specifically interact with one specific antigen. And this interaction between the antigen and the lymphocyte receptor for which it's specific makes these lymphocytes be activated. So an interaction between a foreign antigen and its cell that holds a receptor for which it's specifically activates just that lymphocyte. So in this example, only the central lymphocyte would be activated because this foreign antigen, which is a circle, cannot interact with either of these 2 receptors. Upon these activated specific lymphocytes, so here depicted in yellow, after activation will then proliferate and differentiate into what are called effector cells. And these effector cells act to eliminate their cognate antigen. So as I previously said, lymphocytes before activation have very little active function in the immune system. They only gain a function such that they become effector cells only upon interaction with their cognate antigen. And by cognate antigen, I mean the antigen for which the receptor is specific. After differentiation, these effector cells, so the cells that have been activated, will then bear receptors that are identical to those of the parental cell. So when a cell is activated, it would proliferate such that there's a very large population of cells that's specific for the encountered antigen. This allows for a very large pool of cells that can act to eliminate the specific antigen. So now that we've gone through the general principles of clonal selection hypothesis, or basically the outlines for how the adaptive immune system works, let's dive in a little deeper and get to know T cells and B cells a little bit better. So T cells and B cells can really be looked at from the receptor's point of view. So T cell and B cell receptors are how they interact with their antigens. So antibodies and B cell receptors are synonymous in most cases. So here I'm depicting very schematic illustrations of T and B cells, but before we dive into the specifics of these individual different proteins, I really want to emphasize two very important factors that these two molecules share. First, they have this variable region, which is also called the antigen binding site. So these sites are very critical because these are the sites on T cell receptors and B cell receptors that will actually interact with the antigen for which they're specific. So these are the places that allow for the specificity of the receptor. Our molecules also have constant regions. So these are regions that do not differ between different antibodies and T cell receptors, and they allow and determine the actual functionality of what the antibody or T cell receptor will signal to do. For example, these will signal for the cells to be activated. So just as a reminder, we have our variable region that differs between different T cell receptors and antibodies, and our constant regions, which remain the same between B cells and T cells. The next slide is going to be very busy, and I don't want you to get worried because this is a concept we're not going to be diving into. But before we go into how these two molecules work, I wanted to give you a very brief snapshot into how this variable region is constructed. Because you might be thinking, okay, I understand that different cells have different specificity, but how can that be encoded when we have up to 10 to the 12 different B cells and T cells in our bodies? Well, this occurs because antigen receptor genes are assembled by different systems of somatic gene rearrangements and somatic hypermutation, such that within our genome, we're encoding different chunks of these receptors, which are then assembled in various different ways to lead to variability. The actual concept of this will be covered in a future lecture, but I just wanted to give you a very brief overview that indeed each individual developing B cell and T cell will have a very different specificity when they leave the bone marrow and the thymus. Now let's dive into antibodies. So antibodies look like this. Well, they tend to. They tend to be in the shape of a Y, you can see here. And again, they have those two major regions. So first, here seen in red, we have our variable region. And our variable region is what interacts with the antigen for which this antibody is specific. And we also have the constant region here that's illustrated in blue. Now, if we change the colors a little bit of this antibody to show you a different aspect of antibodies, antibodies are actually made up of two identical, what are called heavy chains, seen here, and two identical light chains, seen here in yellow. So what you can come to appreciate if you look at these two pictures together is that antibodies are heterotetramers, which means they are made up of four different chains, so two heavy and two light. And then the constant region of antibodies is made up of just heavy chain. And then the variable region is a combination of a light chain and a heavy chain. So you can see that the variable region has even more diversity than you would think from just one chain, as it combines two different chains. If we dive into this variable region of the antibody seen here, and we look at the heavy chain and the light chain regions, so V here signals for variable region, and we look at how the antibodies are structured, they are structured with a combination of framework and hypervariable regions. Now, framework regions are what create the structure for the antibody, so these are really important to keep constant and consistent because this allows for the antibody to maintain its shape and to allow for it to have functionality. And then the hypervariable regions, which are here scattered in between the framework regions, seen in the heavy and light chain regions as red. These regions are what give the antibodies very high specificities. So these are regions with very high variability. And you can see by this chart, which looks at the degree of variability, you can see that variability between heavy chains in the framework region is very low and very consistent. But if you look at the high variability regions, they skyrocket, and this allows for a very high specificity. And you can see this type of pattern in both the heavy chain and the light chain. And this shouldn't be surprising, as I mentioned before, as the variable region here is made up of both a heavy chain and a light chain. Now, in terms of how antigens are recognized by antibodies, different antibodies have specificities for different epitopes. Now, an epitope is a certain part of an antigen. It's not the entirety of the antigen itself. For example, as you can see here, we have one specific antigen that has these two epitopes that are being discussed, and we have two antibodies that are specific for these two different epitopes. You can imagine that you can have antibodies for a variety of different other epitopes. For example, this loop here or this loop here. For one specific antigen, a variety of different antibodies can be generated, as different antigens have multiple epitopes. Now, antibody recognition really depends on the structure of what the epitope looks like.